screen. So we're going to look at the direct connectivity between the nucleus of the solitary tract, the caudal region, to the arcuate nucleus in the hypothalamus, the feeding center, right? The hunger center. So how are these connected? Let's start with figure 1.
That's me. Okay. Okay. So they wanted to learn more about the glucose pathway and circuit, and specifically for figure 1, they wanted to figure out what the main source of catecholaminergic, so like catecholamine, which I think is like, isn't that epinephrine, norepinephrine, input for the circuit. So they just basically wanted to figure out where it starts. And so they first wanted to test if it started at the NTS, because that's what usually projects to the hypothalamus or the arc neurons in that hypothalamus. And so they got 1A is showing TH-Cre mice. So they activated the Cre-dependent TD tomato in neurons that express the Cre recombinase so that they could label them. And they did this with an interrograde method. So then it also labeled the arc neurons in the hypothalamus, which is where first order regulatory feeding neurons are located. So that's shown in 1A. And then so the TD tomato is red and then the TH neurons are green. So 1B is showing that both are expressed overlapping in the NTS, and C is showing that same thing, but in the arc, which means that the anterograde activation worked. The NTS TH neurons in D in the anterior section were everywhere, but posteriorly were mostly lateral. So what they got from figure 1 was that in conclusion, NTS is a major source of catecholaminergic input for arc neurons in the hypothalamus. And that's where the circuit basically starts. So that was it.
So why are they looking at TH? Why are they using TH-Cre animals?
That's what I was trying to figure out. I couldn't really find that. So
yeah, I guess my question here was, does, I can't remember, I know, is it dopaminergic neurons that also express or are positive for TH? TH is an enzyme that breaks down, I can't remember what exactly it breaks down. I know it's an enzyme that breaks down neurotransmitter. Can't remember which one. Tyrosine?
Yes.
So is that part of?
Well, TH neurons are generally like a dopaminergic neurons, I think, right? Because they are part of the dopamine pathway. So here,
So I think if you wanted to see the dopaminergic neuron, maybe you can use the V-GLUT2 tree, but they wanted to see the catecholamine thing, so they using the TH tree, I think.
I'm trying to pull up. Are you guys seeing the Google page?
Yes,
Yeah. Okay.
but it's small.
Yeah, it's still small. Oh no, I don't want the YouTube.
I sent a link, which
Oh, you did? Okay.
yeah,
In-
there's
Doesn't
a
this
very
happen
good
in-
image there. Just show like what TH does.
yes.
Doesn't
And there's
catechol,
yeah,
sorry.
the first picture.
Doesn't that occur in the kidney?
Yeah.
But we're talking about the brain.
Also in the brain. Yeah.
Oh, I thought it was just in the
Dopamine is produced in the brain, right? Dopa. So this is
Tyrosine, yeah.
pH is the first step of this. So
Yeah.
For dopamine to be made, norepinephrine, epinephrine to be made, they all require this first step, pH. Right? So when you use the TH-CRE in these neurons that express tyrosine hydroxylase in the NTS, then you're labeling all of them. So you inject in the NTS. What do they say that B is? Why do they call this B? Right. Oh, this is a section of the B. Okay. And section of the C. So they inject in the NTS and look at the projections that go to the arcuate nucleus. Right?
I was just a little bit confused because wouldn't the TH neurons be the ones that are expressing TD tomato, right?
Right.
So they'd appear red, but then why are these other TH neurons green? What's the difference? I think that's where I was getting a little bit confused.
That are green but not red?
Yeah.
Those would be the ones that
It's an antibody labeling they
yeah.
did for TH.
Yes.
It's specific. I was also
Oh, so that's
baffled
just
how
showing that the TH TD tomato
positive.
thing worked.
Yes, they're
Okay.
just showing the TH labeling, that is where are the cells with antibody staining, and then TD tomato is just TD
So
tomato.
in a perfect experiment, you would expect the green and the red to be 100% overlapping.
Okay, that's what I was thought, but okay, got it.
But you do see, well, you would, but practically, when you inject viruses, you would think that not all of them are infected, right? Well, yes, not all TH neurons are infected, so you would see some green cells that are not red. That, I guess, is acceptable, depending on how much virus you injected or where you injected, if you injected in the correct site, right? So that makes sense now? All right, so in C, they look at in the hypothalamus, right? So they see all these red projections that came from the NTS. Here. They do staining again for TH. Just, I don't know why particularly, but they do see some TH cells there, which isn't surprising, but I don't know what this really adds to the figure.
Maybe I think almost TH is projected to the VMH after the ARC. So they have one more time staining the TH again. ARC also project the TH also. They want to do it. They want to see that.
But if the experiment worked well, one thing you could see is, let's say you have an arcuate nucleus TH neuron that projects to the NTS, right? Going the reverse way. Then you could have that virus be picked up by the projections of arcuate nucleus neurons that go to the NTS. And here, by showing that most of the green cells here are not red, then you can conclude that the projections, at least for the TH, is mainly going from NTS to the hypothalamus and not the other way around, right? And this shows the entirety of the arcuate nucleus. Okay. Figure 2? Yes? Figure 1's clear? No questions? So, yeah. Was that John?
Yeah.
Yeah.
Okay. So for figure 2, they wanted to.
Is it just me or is
Oh,
this
no, John, the sound is weird.
Yeah.
Your sound is very weird.
Give me 1 second.
Yeah, you're breaking up a lot.
Wait, can you do that again? We can't hear you. Now we did. Yes.
Oh, okay.
Better. Yes.
So for figure 2, they wanted to do optogenetics with the projection of the TH expressing neurons from the NTS.
Mhm.
So figure A is the diagram or the schematic of the experiment. And then in figure B, they noted, and figure B and C, they noticed that activating these TH neurons will lead to an increase in food consumption. And the amount of activation actually affects the amount of food consumption. So in figure D, they tested activating a specific number of neurons, and the more neurons that were activated led to more food consumption. And figure E and F, they changed the frequency of the.
Let's go a little bit slower.
Okay.
So just to emphasize, they're injecting the virus, the channel rhodopsin virus, in the NTS and then shining light in the arcuate nucleus, right? So that you're stimulating only the neurons that project from the NTS to the arcuate nucleus that are TH, that express TH also.
Yes.
Yeah. Okay, so then let's look at B. Right? They shine light, and what are these? What is each row?
A mouse.
So they're looking at multiple mice, right? Mouse number 1, 2, 3, 4, 5, 6, 7, 8. Is it 8 different mice or 8 different trials of the same mouse?
Let's see. Each row represents an individual mouse, and each tick mark represents a single food pellet consumed.
Mhm.
So it's 8, and I guess 8.
Yeah. So, but I mean, is it striking enough? Is it obvious enough? In a 2-hour period, you're shining light, right? Let's just assume that you just look at the CHR2 mice. If you take them together and you compare the control versus the CHR2 group, then you do see this significant increase during the stimulus, right?
Mhm.
But just from this, it doesn't seem like very striking. Right? Except, I mean, I guess if you compare the blue area versus the white, then you see that there's a tendency of the mice eating while there is blue. And that's what you see here. So what I'm trying to get at is that when you shine light, it's not like the mouse races to the pellets and starts eating like crazy. It's a much more subtle effect.
Do they shine the light for 4 hours straight? Isn't that a lot?
Seems like it. And yeah, so then they also try in different frequencies.
Right. So in E, they tried 5 hertz, 10 hertz, 30 hertz with light stimulation, and they noted that in figure F, the higher frequency led to a higher food consumption.
What are they doing in E? What are these figures showing?
Yeah, I'm confused. Like, what is is this voltage? Is this current? Like, I don't understand what they're showing me. Is this a slice experiment? Like, I'm not sure. All they say is like seal recordings. I don't really know what those are.
Says that E is a representative trace of loose seal recording with light pulses. Oops.
How is this done?
So the question is, what is Lucille recording? Because they did it again somewhere, and I was
So in
confused.
their methods, they were talking about the grain size recording, and they have Lucille recording as well as the GCamp imaging on the grain size, which shows in the figure 6. So presumably the figure 2 should be the same thing. Node patch recording is just a different form of patch clamp recording. It's just like the membrane is still intact. So what is inside the cell is still inside the cell. It will not wash out by what is inside the pipette. You can treat it as a more like real physical conditions of the cell electrophysiology. And what it basically shows is, I think it shows just the fidelity of the genetic simulation with the neuronal activity. So each pulse of the mice corresponding to a single action potential, as shown by each of the line in the E. I think this is what they basically say, but they didn't quantify that. So usually there will be some. So usually, like there's some failure of action potential upon the channel Dobson stimulation. at a higher frequency end. But it seems that they want to point out this work quite well, even with the 30 Hertz simulation.
So this is a depolarizing current. So whenever there's a light pulse, the neuron depolarizes.
It's not current. It should be the voltage change.
Voltage. So they just don't include a scale. So this could be like 5 millivolts for all I know.
Yeah, that's a pity. It should have a scale. I didn't notice this. Should have a scale.
Yes, but so if we go back to how they would do this, is they would take a slice, right? I would imagine they're doing the NTS slice, and they'll target the red cells, right? Because the channel rhodopsin should have like YFP on it, I think. Yeah, so channel rhodopsin has the EYFP fused to the channel rhodopsin. So you can look at the green cells or the yellow cells, yellow fluorescent protein. So they would be patching onto the yellow cells, and then have a optical fiber that would shine blue light at these pulses while they're doing essentially patch clamping. Yeah. So that's what they're measuring here. And in the absence of a scale bar or y-axis, you don't really know what kind of responses these are. But clearly, when there's when blue light is shining, there's a change in voltage. And it does seem like the first few are stronger, but even after 2 seconds, 1 second, you do still see persistent activity. Whether you see the same kind of activity after 2 hours or 4 hours, I don't know. But that's what E is. And then, John, do you want to take it out again? Go over F.
Yeah. So for F, with the various frequencies, they noted that there was a positive correlation between neural activity and the amount of food being consumed. So like a higher frequency, there was more consumption.
Don't know what that means, but
and
yeah.
In G through I, they noted that during the stimulating period, there was an increase in like the frequency of food consumption. So G measures like the delay to the first meal. Figure H shows the size of the food, or like the weight of the food being consumed. And I is again the frequency, so how many times they consume during that period.
But like G also, I find quite surprising, right? Because the graph looks great, but you can see that this is almost 1 hour, like 40 minutes maybe. 40 minutes after the light is started shining is when that's the latency to the first meal. Right? It's not like that rate that these mice race to the food. So.
And then figure J, they moved on from optogenetics to chemogenetic using DREAD. And they used that by using HM3DQ with M-cherry protein. So the schematic is J.
Yeah, so J actually is quite complicated. look at this in detail and what's happening, because they're using two constructs, right?
Yeah, so I don't know if it's overcomplicating or this is the only way that they can make it very specific, but they did it again later. Is this overcomplicating things?
Why would they have to do this? Let's say you injected the DQ that everyone has been, most people here have injected, right? Let's say we use the regular Cree-dependent dio DQ. What happens into the NTS in a TH Cree animal? What happens then?
You label all the TH neurons in the region.
And you'll be stimulating all the TH neurons in the NTS, right? Which the majority probably don't go to the arcuate nucleus.
Yes, some of them go to other places.
Yeah.
We can see it in 1C.
Right. And when you systemically inject the CNO, then you're going to stimulate all those other pathways that they're not focusing on. We don't know what that's going to do, but I would expect that, because most times when you stimulate the caudal NTS, that causes the animal to eat less in most cases, generally speaking. So then I think if we were to repeat this experiment in the caudal NTS, in the TH Cree animal, and inject the Cree GQ, then we would see the animal eating less, not eating more. So then to specifically activate the NTS to arcuate nucleus projecting neurons, they're using this trick of using two recombinases, right? The flip and the Cree.
Yeah.
So then the Cree, so in the NTS, you would have, you guys can draw this out, but you would have this, you'll need the flippo. Right? How does this work? The flippo.
So
Is
flippage will be retrogradely transported back to the NTS, expressed in those neurons.
Mm-hmm.
At the same time, the NTS neurons also express F-diol, so when they combine, they will trigger the DQ expression. That's how it works.
Yeah, so the black and the white triangles are Cree dependent, right? LOXP targets. And then the purple is the FRT site. The part that I'm trying to draw out in my head is the F-diol.
It should be controlled, right?
That is also Cree dependent, right? So what happens if?
yes.
Well, it should be.
So it. I'm trying to draw this out. Oh yeah, in the end you need both, right? For the DQ to express.
So EF1A is a Cree dependent.
Cree dependent.
Right? So that means the EF1A in NTS.
No. EF1A is not Cree dependent.
But the arrows are black.
Both
and then
of them
what's
are
gonna
EF1A is a elongation factor which is expressed everywhere. So the
yes,
promoter
but you need to
promoter is a very strong promoter.
Right.
All cells express this at very high levels, right?
So here
but you need to activate it with So you need to flip it with
Mhm.
pre.
Mhm.
And then when that's active, it goes back to NTS.
Mhm.
It flips with the flip phase, the diode, the DQ, and then it goes back
Right.
to ARC. So you do need the two different systems to work.
So the black arrows here make sure that flip is only expressed in NTS.
In Cree.
Mhm.
Or like in Cree, right?
Because it's TH Cree. And we know there's TH Cree
Right.
cells in the ARC.
But then make sure NTS TH neurons express the flip, and the flip will control the F dial.
So in the end, these TH neurons in the NTS will have both constructs, right? They'll eventually end up in the same place. And I know that like Amanda definitely, and we've looked at this dial and how this, after two flipping arounds, ends up being permanently in the right orientation. Right?
To finish out the figure, figure K is showing some CFOS activity overlapping with the DQ receptors that are expressed within cherry. And then figure L is shows the CFOS activity when the agonist or when it was injected with either saline, a control group, or the CNO.
Mm-hmm.
And then in figure M, it just shows that similar to optogenetics, activating these same neuron projective leads to an increase in food consumption.
Yeah. I mean, it's kind of the same thing as the optogenetics. Yeah. Doesn't really add much information. Yeah. We're going a bit slow with the figures today. All right. So figure 3.
Okay. I'll just go really quick. So figure 3 is a continuation of figure 2. Here they are just using the inhibitory chemogenetic manipulation. So they're using the HM4DI here to inhibit those neurons. So the setup mechanism is the same. Express flip in NTS combined with the F dial. So that's the HM4DI will be expressed in the ARC projecting neurons. And B is just testing the firing rate of these neurons. We can see upon CNO injection, the firing rate just decreased dramatically after about 2 minutes. And C is just a quantitative frequency measurement. So that upon CNO injection, the frequency is much lower. In D, they're testing that if this inhibition of these NTS neurons manipulates or changes anything for the fasted or hunger driving feeding behavior. And they find there's no significant differences. We can see in the black and the red bars, there's no significant differences, meaning that the food deprivation driven feeding behavior is actually not changed. Therefore, they tested that it is specific to like a glucoprivic. Is it glucoprivic? Mm-hmm. Glucoprivic feeding behaviors. Mm-hmm. So they have well-fed mice in E, and then they are comparing. So here, in addition to saline CNO, they also added 2DG, which is a drug that will trigger this kind of feeding behavior. So we can see in the control group on the left, when you inject EYFP, upon application of 2DG, you have significant increased food intake. So if we compare the gray bar versus the orange bar here, So that means it's driving the feeding of the mice. However, in the HM4DI case, if we compare the two orange bars here, we can see that behavior is decreased by the chemogenetic inhibition. However, the yellow bars and the control group are not changed much. So here they are just concluding that it is a major contributor of hypoglycemic hunger. So it's all about glucose. So it's specific to glucose driven. Yeah.
So how does this work, the 2DG? How does this induce glucoprivic?
Does it just decrease the glucose level?
Because what is 2DG?
Deoxy D-glucose.
Oh, so does it prevent the glucose to go through further glycolysis because it's missing the oxygen molecule?
Mm-hmm. That's right. So then there's, by injecting the 2DG, you're getting less glucose in the body, right? And that induces this glucoprivic hunger. So there's increased food intake. If we try to psychoanalyze the researchers here, right? You would expect from until the previous figures that if you inhibit the activity of these NTS neurons that project to the arcuate nucleus, then perhaps you're going to see this effect that you see in E just by fasting the animals, right? But that they don't really see that. They have to go through this specific glucoprivic pathway to see an effect of inhibiting these neurons. Yeah, can we move on? Makes sense so far? Everything's making sense?
Yeah.
Right. So in figure 4, what are they doing in figure 4?
Okay, I think let me quickly go through it, save the time. And first of all, I don't know why they want to do this. And this is another look at previous slides talking about the CNO effects, local CNO infusion in acute nucleus, and for the inhibitory drug, it reduced the food intake. Yeah, I think so. And then, so they pick up the same similar strategy using the optogenetics, starting from NTS, and then they look at its terminals in the ARC. Again, at the bottom part of the A, you see basically this is the what do you really see with the brain size when you doing the patch pan recording with brain slices. And then they start with the B. You have the stimulation on blue light, 10 hertz, and with about maybe 20 to 30 seconds. And you will see there's example of traces from the top to bottom. You have example where the neuron activated constantly during the stimulation period. But you also have neurons inhibited, like in the middle. And you also have neurons that doesn't feel anything at the bottom. So they do a quantification. Most of them actually doesn't change. That's a surprise, right? If you go back to the behavior effects, I think in the figure 2. And then what does these, so they further categorize the in D and E, right? You have, sorry, I'm not sure what the BL means. Probably means baseline. So anyway, it shows, quantifies us like the effects of blue light stimulation. So we focus on the difference between the blue bar versus the black bar, left and right, blue bar, black bar. So basically stimulation works, increase the frequency, and in D and in E, I think it's baseline. So percentage of baseline. Right?
Baseline, yeah.
So like blue bar here is should be 100%. So that's okay. And then in E is a little bit complex. They are adding different, you see those, all those acronyms. Those are different drugs targeting different membrane receptors. I don't know what PRA is. Oh, sorry, I do. So in E, there is the alpha-1 adrenergic receptor blocker. Probably those receptors expressed in the ARC neurons. And it seems that this, oh, okay, they tell us that it doesn't, it's not this increase of frequency doesn't come from the, doesn't come from the adrenergic receptor, but it does relates to the glutamatergic transmission. So if you block it, because they stimulate the axon, so presumably stimulate the axon, you should release some of the whatever transmitters inside that axon, and basically with the CNQX and AP5, they block these glutamatergic receptor either mostly likely is in the soma, so in the ARC neurons, not in the NTS axon. That's my hypothesis, because those mostly are AMPA receptor block, AMPA receptor and MDA receptor antagonist. So far it's good. They just tell us that so far it tells us they NTS sends glutamatergic input to the ARC neurons, but interestingly, most of those inputs has, like say, doesn't do anything to the ARC neurons because they didn't evoke any firing changes, summarized in figure C, I think. So in F, what they do? I think they're doing like a local, in F. So they do a little bit of modification on the same paradigms, injecting NTS with the Channel Dobson virus, and at the axon of as its axon targets in the ARC, they do both with the laser shining laser, but also they try to further using the PRV blocker locally infused in the ARC. And this time, well, I think it's not like it's not the PRA. It's a different.
It's the alpha 2.
Yeah.
Yohimbin.
Yeah. It's alpha 2, so
Yeah.
it's another blocker.
Yeah, it's another blocker.
Wow. So
it's a cocktail. Yeah.
So it's a cocktail, and somehow it works block out the like a glutamagic trans. So you will assume in G in F and G, like with upon the laser shining, there's still glutamagic input to the ARC neurons, and ARC neurons do have the glutamagic current, so they should increase. But it seems that the adrenergic receptor also plays a role there, because it's completely block the NTS effects. So what's going on there? Maybe because of this cocktail block the glutamagic transmission altogether at the presynaptic level. Maybe because of the postsynaptic effects. I don't know how that.
Yeah, so if you spend if you put in enough effort into trying to figure out what's going on in this paper, you might have found that the paper, especially the legend, is so poorly written because there's not enough information there to figure out what's going on, right? So yeah, I would blame the paper and the authors for it. But also the way that the logic here is kind of it's so loose, right? Because, for example, we know very well that when you're treating multiple things, you want to show like effects of individual ones, right? And here they're trying to push the fact that that adrenergic receptors are doing something, right? Not very obvious from E, maybe from G, right? But overall, I think the evidence that's supporting that adrenergic receptors are doing something is a maybe to me. It's not so clear for me. Right. What do you guys think?
I hope they have in E. They try to. I hope in E they could show us the effects of adrenergic cocktail, the effects of it.
Yeah. Should we move on? Figure 5. So I think the types, let's see. So they look at the activity, right? So they do the electrophysiology again, and they would characterize this arcuate nucleus neurons as, oh, this one increased activity, this one decreased activity, this one had no activity, right? So then they would, from that pipette, they would take up the cell or the contents of the cell, and then they do RT-PCR on it to see which markers are expressed, which arcuate nucleus markers are expressed in that particular neuron. The main ones being the AGRP neurons and the POMC neurons. Yeah. So they are saying that the majority of the ones that showed increased activity are AGRP positive. The majority of those showing decreased activity are POMC positive. Right?
It means they did pick up the cell in the ARC.
Mm-hmm. Yeah, they're picking up the cells in the ARC. Which it just seems so forced for me, because what they're trying to say is that the connectivity of the NTS neurons to the arcuate nucleus that induce activity in the arcuate nucleus. So the cells taking up signal in the arcuate nucleus that get the signal from the NTS are AGRP positive, which are known to be hunger inducing, right? Right.
They can do a self-profiling here. It's way more convincing than design, like designate some markers and look for evidence for them.
Mm-hmm. Yeah.
I think one of the weaknesses of this figure is the very small portion of cells that's increased activity and also decreased activity. It's like
In
9-10.
bigger. You mean these? Yeah, they're looking at.
Yeah, they also detail that in the legend for the next figure. So they tell you for B, it only shows like 12 cells in 100 over 110 neurons, 10 mice.
Yeah.
And what happened to those, unchanged neurons? They must have been sequenced, not they did any PCR, and then how much of them shows up as HRP? How much of them shows up as POM, POMC?
Yeah.
And they're trying to make a point that is consistent with the idea of HRP in the feeding, its role in the feeding behavior.
Yeah.
And consider the TH neuron except the HRP neurons. So in that sense, theoretically it makes sense.
Mm-hmm.
But there's a large amount of the unreported, I think, kind of unreported neurons that doesn't show changes, and I don't know.
Yeah, I just feel like the last couple of figures are so forced based on what we know about the arcuate nucleus. So they just try to fit this into what people already know about the hypothalamus. It just felt so forced to me, and the data is not as strong. That's what they would like to conclude.
And C is for 4 neurons.
Yeah.
So you can see what the percentage really means to you.
Yeah, yeah.
It's,
And this probably is something that's already known, right? The HRP neurons and the effect of the blocking HRP neurons in the glucoprotein hunger. So we'll move on. So figure 6. So what are they doing here?
so they were using a different way to characterize the MPS input to ARC. But this time they use a different mouse model, including the MPY GFP neurons, because ARC also expressed the ARC is heterogeneous with different cell populations. MPY is one of them. I think TH also is one of them. So all they using the same similar channel dopsin virus in the MPS, shiny light in B and C. You see how you have connections in C, like exemplary connections. And you also have cases that show no connections, as in B. So B and C shows the current is postsynaptic. I think before the light shining on in C, for example, those downward bars, those are like inward currents. And these are from spontaneous activity when there's no simulation. Yeah. And then when simulation on, there's also some of the downwards inward currents, and D is more clear, is clear because it shows like basically have no effects. And then
so MPY are probably the HRP neurons use MPY, right?
Oh,
so then what they're doing is in slices, they're stimulating by light the NTS or the fibers originating from NTS, and then doing patch clamping in the HRP neurons. And this A to D seems to show that even when you stimulate the fibers, you're not directly activating the HRP neurons. So there's no direct connectivity between the neurons that come from the NTS to the HRP neurons. Is somebody saying something? No? But when they just squirt norepinephrine onto that slice, and in these cells that they're patching, then they see activity. Right? And same with imaging, if they do calcium imaging on the HRP neurons and put norepinephrine, they're seeing activity. So then the question is, like, where is this coming from? Where is norepinephrine coming from if this is used to activate the HRP neurons, not directly from the NTS neurons. So let's move on to the next figure. Should we just briefly go over this figure? I think we all know that the main projection to the NTS is goes up to the brain through the PBN, right? So here they're trying to see if it's the same neuron going to the PBN and the arcuate nucleus, or is it separate populations? So then they put the green bead in the PBN, green retro bead in the PBN, red retro bead in the arcuate nucleus, and then they want to see the overlap, right? In the TH background also. So they're looking at green coming from PBN, red coming from the arcuate nucleus, and then TH antibody in blue, or far red in blue. And this is the pie chart that they get. So this would suggest that most of the TH neurons in the NTS are going to the arcuate nucleus, or not, 59%. No, actually more, right? Because there's 15% that goes to both. What was this again? I guess here they mark the neurons. They mark the TH neurons that project to the arcuate nucleus, and then they do again electrophysiology here while they're presenting ghrelin, which is a stop feeding signal that comes from the intestine. I don't know what to conclude from this. You lower the glucose level in the bath, and then you see like half goes up, half goes down. Yeah. And a very long section on limitations of the study. So first received in November 2018, revised 1 year later. So I find the connectivity interesting. I don't know what else we can learn from it, that perhaps it promotes feeding, but the effects are not very obvious. It's not, it'll have to be a subtle change that you observe. Okay. Anything else about the paper? We're 15 minutes over time, so we have to rush through the last couple of bits. We good? Okay. All right then. I'll see you on Tuesday.
Yeah.
Bye.